Good afternoon, ladies and gentlemen. We are 2013 iGEM team Tsinghua-A, and today I'm very glad to be here to present our project. Synthetic gene switch shows adaptation to DNA copy number variation. Well, it seems a little bit confusing to understand such a gene switch and its function. But don't worry, let's get a glance of our ideas through a little story. There once exists a cell kingdom, and the cells there live a happy and peaceful life. But recently, the cell citizens noticed that there are some dark forces in the whole country, and they have already made some destructions to the kingdom. In this kingdom, the ordinary cell citizens have red hearts, but these evil cells have black hearts. But they have a strong disguise and hide around the ordinary cell citizens. But they may suddenly approach the ordinary citizens and attack them. Therefore, these ordinary cell citizens will be turned into evil ones. So, the dark force is getting stronger and stronger. This dangerous situation really worries the king. So he commands all the police to search for the evil cells. But since the evil cells look exactly the same as ordinary ones from outside, it's really difficult for the police to figure out the different kinds of cells. So the king is really worried and disappointed. However, luckily, a group of cells from Tsinghua University claim that they are trying to figure out a new way to tell which one belongs to ordinary cell citizens and which are the evil cells. The king is so excited, and he hopes that with the help of this new method and device, the evil cells can be recognized easily without mistakes. And after all the evil cells are caught and killed, and the life will turn back into normal of the whole society and the kingdom. So what is the secret of this method? And how could it be smart enough and reliable enough? Well, let's get closer to today's Tsinghua-A's project. Our project is originated from the cancer treatment. Well, any specific therapy to kill cancer cells greatly demands a method to recognize cancer cells. For example, in HeLa cell, a cancer cell, the amount of a certain kind of microRNA 21 is very high, but the amount of this microRNA in ordinary cells is rather low. This remarkable difference inspired us to design a gene regulatory network, of which the output is related to the input signal of microRNA 21. So this kind of design can sense the difference between low input and high input. Therefore, the cancer cells can be recognized, and the ordinary cells would be safe enough to remain in your body. But we noticed that there are some complex factors that may arouse noise during the gene expression. For example, the number of DNA template, also known as DNA copy number, may have a strong influence on the final expression of protein. And actually, the exact number of the DNA templates is very difficult to measure or control precisely. So it's very important to introduce the character adaptation to DNA copy number as a key character of your gene regulatory network. And here, the adaptation is defined as the ability of a network to function robustly and keep the same level of output despite the fluctuation of DNA copy number. So our goal is to search for a gene regulatory network that can sense the input signal and realize the switch function, also show adaptation to DNA copy number variation. And now I'd like to introduce the whole process of our design. And firstly, we did some modeling work, and secondly, we carried out some wet lab work to verify our hypothesis. And finally, we came to the conclusion. In our project, the gene regulatory networks are abstracted as meshed networks with nodes representing genes and the edges representing the relationship of regulation. And there are about 20,000 networks here. And the kinetics of the regulatory H is described by Hill equation. Here, the function inhibition and the function activation. And the whole network is described by the ODE sets here. First part brings in the mutual activation. Second part, mutual inhibition. And the third part, self-decomposition. And in our project, we fixed two edges here. The input inhibits the A node, and the A node inhibits output. This is because when we carry out the wet lab work, we find out that the components which act as inducers are quite few. So we use two repressors to substitute it, which is easier to be operated. And as I have mentioned before, our target network is supposed to be able to sense the difference between low input and high input. So it should have the kind of input-output character like this. Low input, low output, and high input, high output. And also the interrooms region should be as narrow as possible to avoid the ambiguous result. And here the two characters are described by two indexes here, high-low ratio and interroom slope. We enumerated all the possible three-node networks and illustrate the result in this two-dimensional map. Here the axes are high-low ratio and interroom slope. And we noticed that the functional networks should be located in the top right corner here at region C, because the interroom slope and the high-low ratio are both at a very high level here. And this is the result of simulation. We screened 476 networks out of the about 20,000 networks as the first round of screening. And the networks are able to achieve the function as a good switch. And the whole process involves massive calculation and parallel data processing. So we finished the first round of screening with the help of HPCC, short for High-Speed Computer Cluster in Tsinghua University. Besides the basic function to achieve a switch function, we pay attention to the DNA copy number as a primary source of noise here. And when we upsweep the DNA copy number as a parameter, the input and output curves should be closer and closer. And when we analyze the relationship between output and copy number, the ideal networks, the adaptive networks, should tend to be saturated. Otherwise, the infinite output of the network will change linearly depending on the DNA copy number, which is what we want to avoid. And we repeatedly solved the ODE sets several times and under different DNA copy number, and finally got the result and tried to measure how soon the output achieves saturation. And here we use the index response value defined as the value of DNA copy number when the output ultimately ended up within an error range of 2%. And this is the simulation result. We notice that the yellow curves shows no saturation, and the blue curves show saturation with the highest speed. So we selected about 111 networks out of the 476 networks at the second round of selection. And these 111 networks are supposed to be able to achieve the function of adaptation to DNA copy number variation. And interestingly, we find out that all the networks that are adaptive contain some certain kinds of motifs here. This is a basic stem, and others can be classified into groups as mutual inhibition, feed forward with a buffering node, feedback with a buffering node, and self-activation, also a double effect here. So we ranked all the networks, the adaptive networks, by their response value, and classify them into 10 categories. Each category represents networks containing certain motifs, as I just mentioned before. This paragraph shows that the appearing frequency of each core motifs, and it's clear that they feed forward IBA appears most frequently, and also the purple curve helps to indicate that they feedback this mutual inhibition structure AO may also play an important role in showing adaptation to DNA copy number variation. So we came to the hypothesis that these two core motifs may have significant influence on showing adaptation. And considering that actually the number of DNA templates that are transferred into cells is not at a certain amount, I mean, always it follows some distribution of probability. So we assume that the DNA copy number may follow the Poisson distribution, and we study the distribution of output when input is either low or high respectively. And we calculated the overlap region. And it is clear that the smaller the region is, the better the network is. So this is the simulation result. This is the result of an ordinary network. And this one is for an adaptive network. And it is clear that this region of net overlap is much smaller than this one. And for further analysis, we introduce a sorter case simulation. Here we offer a certain amount of cell A and with low input, and cell B, the same amount of cell B of high input. And we calculated their overlap region of their output distribution, and use this to calculate an index called accuracy rate. And we find out that only adaptive networks can distinguish red and green cells, I mean the cell A and the cell B, with high accuracy rate, but the ordinary cells cannot be smart enough to do this kind of stuff. And with, after all the modeling work, let me remind you of the two networks, the motifs, as our hypothesis that they may be able to achieve both switch and adaptation function, and we try to verify our hypothesis in our wet lab work. And now let's welcome our team members to do this kind of introduction.
Good afternoon, everybody. I'm very glad to introduce you our wet lab work. As you can see, we have done a lot of modeling work, and the result was over there. And we pick out a few candidates. So the next step we're going to do is to design a set of specific circuits at the implementation of those special motifs, and to testify the result of our model to see whether they are correct or not. So the structures we pick out is here. So why we chose these structures? Because it is very time-consuming to artificially construct a set of gene regulatory networks, because each network consists of many plasmids. So before we start, we first read a lot of previously published literature and trying to find something helpful. And so what we found is such a substructure is called, we call it feed-forward with buffer node. It is proved by previous researchers that such a substructure shows very nice switch property, which can be used as gene switch, as we mentioned. So we went back and checked our model and found that this congruent, which came from the experiment, is exactly the same as what our model predicted, which means this result is coherent with our model. So based on this congruent, we then trying to figure out another kind of feature, which is adaptation ability. Remember what we cared in our project. According to our model, such a substructure, the mutual inhibition, which means the two nodes inhibit each other, it has very good adaptation ability to DNA copy number variation. So we combine these two substructures together and get the network number 1. And for network number 2, the only difference is that there isn't a negative feedback edge, so leading to a contrast between these two structures. And as for this structure, there's no input. We can see the input node cannot interact with other nodes, so it can be used as a contrast to testify these structures function. And here are some informations about our plasmids. we constructed all the plasmids by ourselves, and all this information can be found on our wiki. Besides, we conducted our experiment in mammalian cell, in HeLa cell, as we mentioned. It's kind of, I think, very famous cancer cell. And let's move on to see the details about our circuit. So here is the circuit number 1 we constructed. It is corresponding to the structure of the network number 1. Here we can see that we used the microRNA-21 as the input, which is endogenous in HeLa cell. And there are T21 targets on these plasmids, so that they can be repressed by microRNA-21. And the TRE promoter is induced by RTTA and DOX. And together they serve as an auxiliary node. And here the Lakai protein is produced and inhibit the CAGOP promoter. And at the downstream of this promoter, we used enhanced yellow fluorescent protein as the output. We choose this kind of fluorescent protein simply because it is easy to be measured and examined. And besides, here is a FF3 target, so that it can be repressed by the microRNA FF3. A negative feedback edge is constructed here. And here is the circuit number 2. It is almost the same as the circuit number 1, but the only difference is that there isn't a FF3 target, so there isn't a negative feedback edge, so leading to a contrast. As for the network number 3, we can see that we used the target FF5 to take the place of the target T21. So the microRNA-21 cannot interact with those plasmids, so there's no input. And let's see the result. And here is the result. Here we took advantage of another kind of fluorescent protein, MKT. According to previous published literature, this kind of protein, the strength of its fluorescence depends linearly on the number of DNA copy number transfected into cells. Because we think our tolerance level may be influenced by many potential fluctuating parameters. So we think it's more legitimately to use a normalized quotient instead of directly use the value of EVFP. So here's the result. This axis is a quotient, which means the result is normalized. And as we can see, the average expression strength, the output of circuit 1 is the strongest, of course, because here is a mutual inhibition structure. And the circuit 2 is lower, and circuit 3 is the lowest. So from this pretest, we know that our circuit was successfully constructed. And then here is a result of adaptation ability. And here, facing with the difficulty of counting the DNA copy number directly, we again employed MKT as a reference. We think the DNA copy number is high when the expressed intensity of MKT is high. So here we can see that with the increase of the DNA copy number, the output of the circuit, 1 reaches saturation fast. This red curve corresponding to this structure, the network number 1. And as for the output of circuit number 2, we can see that the line looks like a straight line, goes all the way up. There's no saturation. So we can come to a conclusion that the substructure of mutual inhibition improves the adaptation ability of gene regulatory networks to DNA copy number variation. So that's all about our wet lab work.
After the modeling work and the final wet lab work, we came to our conclusion that we finally got 2 core motifs that may be very important to achieve the adaptation function of a network. And also this can be regarded as a biomachine to sense signals and realize a switch function. And we hope that in the future, this may be used to develop some cancer cell therapies that can recognize cancer cells from ordinary cells and then kill the cancer cells with specificity. And this can also help to refine a gene regulatory network method to synthesize adaptive networks with regulatory motifs. And that's all about our project. And this year we have done some human practice work. And let's welcome our team member to introduce it.
So as for human practice first, our team has done a lot to promote iGEM high school division in China. Our team went to Haidian Foreign Language Experimental School, which is the name of a high school in our city. We gave a talk about synthetic biology and iGEM competition and attracted over 300 high school students. Through this talk, we really got some beneficial and helpful interaction and communication with those high school students. Our team member has also participated in experience sharing seminar that held in Nanjing University. Our team member exchanged their experience doing synthetic biology research with many other Chinese iGEM team. And we also collaborated with Nanjing University as so many of our team members is major in computer science and electronic engineering. So we are very good at setting up equations and coding. So we helped them to build up their model and wrote a MATLAB program for them. And based on our work, they finally got the results they need.
Okay, finally, I'd like to introduce our team members. We have two instructors and 2 advisors. Also, all of the team members are very happy to participate in this whole jamboree. Thank you very much.